Good evening, everybody. My name is Gil Moore, and I'm the director of the CS Mott Center for Human Development, and also the host of Talking to the Expert at the Mott Center. Today, we have a fascinating subject for discussion that is men matter, how the environment affects male reproduction and their offspring. It's a pleasure to have as the expert tonight Dr. Rick Pilsner. Thank you so much, Rick, for joining us this evening. And I would like to start the conversation if you tell us a little about yourself.
Sure, absolutely. Well, first of all, I want to thank Dr. Moore and the Mott Center for inviting me and giving me a job as well. So it's always very important. Just a little academic history for me. I did my bachelor's at Hamline University in St. Paul, Minnesota. It's a small liberal arts school where I studied wolf behavior. I took some time off of school and decided to get back into the environment and received a master's in public health and Ph.D. at Columbia University in New York City. And then I did a postdoc at University of Michigan for 3 years until I got my first faculty position at the University of Amherst in Massachusetts. University of Massachusetts in Amherst.
Okay.
Yes.
And you're back to Michigan.
I am back in Michigan.
And we are delighted to have you with us.
Thank you very much.
We know that this area of male fertility is extremely important, especially in an area like in Detroit. So let's go to the subject.
Absolutely.
So I was fascinated by the title that you give us, Men Matter. I always thought that we have no role. So tell me a little about what we are important for.
Right. Well, I think it's very important for people to know it takes two to tango, right? To produce a healthy offspring, the female is certainly an important component of that. The female is carrying the baby for 9.5 months. And so there's been a really overemphasis on female reproductive health, while really the male has been in the back seat.
And I say, so let me interrupt you in the consensus, because I'm fascinated. You have such a wide range of interest scientifically. What really attracts you to dedicate your academic line and your research on male reproduction? Why are you interested in male reproduction?
That's a great question, and I just want to let everyone know that I have no formal training in reproductive health, and my career is really because of my wife. It was during my postdoc, and we were thinking about having children. At that time, my research was interested in examining the environmental effects of women during pregnancy, so looking at mother cord cohorts. And it's always very difficult to be married to a toxicologist.
So you wanted a toxicologist?
No, not
you wanted a toxicologist. Okay,
right. So we were thinking about having kids, and we had a conversation of, okay, if we're going to be pregnant soon, you should really avoid pesticides. You don't smoke, that's great. Drink in moderation. And my wife is from Long Island, and she's a strong woman from Long Island, and she did not miss a beat. And she said, "Well, what about you?" And that was my reaction.
Okay, you can read, you can smoke, it doesn't matter. Yeah.
I mean, once my sperm does its job, I'm hands off, right? And so it was maybe a week or so later, I had some downtime in my office, and I was just, looking at some research articles and realized that there's been little investigation in terms of environmental impacts for the male and reproductive health, and especially through sperm epigenetics. And we'll get into what epigenetics is in a little bit, but this whole career line of research is because of my wife.
Fascinating. We have to thank your wife and to bring in exactly the importance but we will talk about how the male is. So Absolutely. Why don't we start from a little going basics in order to understand what is the importance of the male in the reproduction, in addition to just donating the sperm and the pregnancy and so on. So would you mind to introduce us to the subject of how the sperm is created? Let's put it in this word. What you will call in medical terms spermatogenesis. Could you tell us a little what is that?
Sure. I think this is a really important component to really understand what my research is really focusing on, which is the preconception environmental health and how that could have long-lasting or an environmental legacy to offspring. And so I have a quick little slide that I brought up with the spermatogenesis. And it's, before I studied sperm, I thought sperm was just what we know of a sperm as a tail. But during spermatogenesis, go back 2 slides, please. No, you're right. That's it. Yes. So what we see, on the left-hand panel, we have a image of the testes and the epididymis, which we'll get into in a second. In the middle portion, we see the different cell types, right?
So we are seeing here is a section of the testicles.
That's right.
What is the reproductive organ of the male?
Exactly.
That we compare it, we say will be in the woman, the ovary.
Right.
The
ovaries, yes. And within that, you have the oocytes.
Okay.
Yes.
So the ovaries, the oocytes, the testicle, the sperm, and the male.
Exactly. And so what's really interesting is the whole process of spermatogenesis, it takes about 74 days. So this is a long developmental window that there's a lot of molecular and different changes that occur during this time.
When in the female is the 30 days that you have every time of ovulation.
Right.
So you say 74 days to get a mature sperm.
Right.
Wow.
And so just some fun sperm factoids. We know that men produce sperm, start of puberty, all the way throughout life. Still produce sperm at 90 years old. Never stop. It may slow down, certainly at a certain age. There's about 30 million sperm that reach maturity daily.
So that is a major difference with a female. So where a woman only will have one oocyte mature, 30 million sperms we are producing daily. Wow, so it really is a machine, it's a factory to produce sperm. 30 million is a lot of cells.
That's right. And it's important to remember that women are born with the number of oocytes. They cannot make any more eggs, whereas the male can keep on regenerating. It's a self-renewal, what we consider the self-renewal of spermatogonia and sperm. One thing I really want to point out is within the 74-day window of development, 14 of those days are in the epididymis.
Okay, so you have to walk us a little because epididymis sounds nice, but I don't think everybody will even know where it is located. Right. Exactly. Alisa will help us to point where is the epididymis. It's there, yeah?
Right. So roughly about 60 days is when the sperm is being produced in the testes. After that, it leaves the testes and goes into the epididymis, and that's where it matures for 14 days, and that's where sperm actually get their reproductive capacity. They gain their motility. There's a lot of different things going on in the epididymis, and so that's a component of the male reproduction, spermatogenesis. People don't think about it. They think that it's just the testes.
Yeah,
but
exactly.
But this epididymis maturation is key to gain motility.
The epididymis, in a way, is like the school where you are inducing the maturation of those boys.
That's right.
To do later the job that they have to do.
That's right. That's right. They walk into that classroom.
They go to the classroom,
but they learn what they need to do and how to kind of protect themselves.
Do they acquire the motility already in the epididymis, or is before that?
That's where they require.
That's where they acquire.
Yes,
so it's a really important step.
Absolutely.
And the health of the epididymis.
Absolutely. There's some things that occur post ejaculation that there's called hyperactivation that occurs in the female reproductive tract. So
maybe we can discuss later when we connect how the sperm communicates with reproductive organ in the female. Exactly. Fascinating. So and again, in terms of the maturation, we did the testes. you have a stem cells, or what is the process that is happening there?
Right. So that's listed as the spermatogonia. Those are the stem cells, and they go through a process called mitosis, and then
And the mitosis means what?
The cell divides into two. And what's really interesting about spermatogenesis, and we'll get into this when we talk about aging, is those two cells that are divided. One is self-renewed, so it goes back to that stem cell population, while the other one is committed to produce sperm. So you have the mitosis, and then it goes through 2 rounds of meiotic division.
Now you're putting as another difficult word.
I tell you. Biology 101.
Oh my goodness, exactly. So the meiosis, what is the end point of this meiosis?
So remember that most cells are diploid. They carry 2 copies of our genome, one from your mother, one from your father. For sperm, for germ cells, you only want half of that genome.
That's when you delete the maternal component. Well, no, let's say half of the component.
Right, half of not. It's
to do the right contribution.
It's not divided to a mom component and a female.
It's not a selective. It's just decreasing the half.
That's right. What's really interesting to know is one spermatogonia produces 4 sperm.
4 sperm.
So it's very, very efficient in that.
And the reservoir is interesting. That is the reason a man can continue producing for years to come.
Yes, the self-renewal. Right, exactly. So you have this dedicated population of stem cells.
Now let me ask you a question, jumping, because why do we need so many sperms? Why 30 million when the one evolutionary? You know, I'm an evolutionary guy, so I always jump to evolution. Why nature will make the male to produce so many sperms, and then the female only 1 or 2, depending on the species?
Right. That is a great question. We only need 1, right?
Exactly. At the end of the day, there's only 1.
Right.
Yeah.
But this kind of gets into sperm quality, which we'll talk a little bit about. Is most of the sperm that we produce, humans are very inefficient at producing very good quality sperm. So a lot of the sperm that we do produce are just not equipped to fertilize or even reach the oocyte.
So you really need to produce a lot in order to have 1 successful, like in life.
Well, you'll have to
running.
There still is a healthy competition.
The healthy competition.
But yes, certainly there's not 20 million sperm competing for the 1 oocyte. A lot of them are not making it up into the female reproductive tract.
So going still back to, maybe I don't know if you still need it. There is another organ that all of us always hear about, and especially when you start aging like me, reaching an age that we are concerned, the prostate.
Right.
What is the prostate?
Right. So I think there's a lot of confusion. I've had people, undergrads, ask this question. They were thinking that maybe the prostate is part of the testes or not. If you're an older gentleman, you've had a medical or prostate exam, you certainly probably realize that the prostate is not located in the testes.
The testicles.
But yeah, it's a small golf ball sized organ that's situated really between the bladder and the penis, and the urethra goes down through the prostate, and so it's important in terms of prostate health. Those people that have enlarged prostates, it kind of constricts their urination and have some problems with that.
And it has one of the main sources of problems in men, that's prostate cancer.
That's right.
Which I hope we can talk about that in one of our future meetings.
Yeah, that is not my specialty, but certainly it is important.
It is important.
But I think another neglected part of the prostate that people don't think about is its contribution to seminal plasma. We talk about sperm, right? And if you think about the entire ejaculate, the sperm only comprise of 5 to 10% of all the fluid. So it's the sperm component is very small.
So much
proportion.
of the ejaculation is made of the liquid that's coming
from liquid from secondary sex organs, such as the prostate and the seminal vesicles. Together, the prostate and the seminal vesicles, which the seminal vesicles are located right near the prostate, they contribute to about 90% of the ejaculate.
Okay.
So people think the ejaculate is all sperm, but it's
It is
only a
not.
small fraction.
Impressive. And do we know what is the role of the semen or in of all this liquid that is contributed to the prostate? What is its role in terms of the sperm?
Right. It actually provides a conditioning for the sperm. It protects sperm
Protection.
against oxidative stress. It also primes the female reproductive tract. It has inflammatory markers. And I think one of the most interesting components of the seminal fluid, it contains what is called extracellular vesicles.
Wow.
Yes,
I think you lost almost all of us, if not a few of the experts.
Oh,
Extracellular vesicles.
What
language is that?
It's still English.
It's still in English. We're still in English. Okay.
But
so tell us a little. What is extracellular vesicles?
So we've identified these for many decades, but we never really knew their function. They're very small components of cells that are released, and they provide cell-to-cell communication. So with the prostate and the seminal plasma, it's a way for the prostate and the seminal vesicles to communicate with the sperm. So these extracellular vesicles can be released by these organs and interact, and these extracellular vesicles contain important cargo, proteins and RNA that provide signals to the sperm to say, hey, everybody's healthy, the environment's good, let's make
like a checkpoint.
It certainly is. It's one of the last steps for the body to kind of interact and provide a signal in the sperm that could influence reproductive
And I suppose
health.
also it's going to have an effect in the female reproductive tract.
Absolutely.
Which I hope we will have time to discuss that aspect.
Sure.
But I see that we're getting a few questions from the public. You want to talk now about the age, or we leave a little later about the age? I think there is a question that maybe isn't in relation to what we're talking. Sperm counts have dropped precipitously since 1950. What are the major causes of this? Is phytoestrogen consumption to be a cause, and how large?
Right. So there was a recent meta-analysis published, I believe, about 2-3 years ago, that certainly in the last 50 years, there's been almost a 50 or 60% drop in sperm concentration in Western developed countries. So this is a dramatic drop in sperm number, and
which will impact also in the success of getting pregnant women.
Right. Yes, absolutely. And what is driving this? We don't really know. But you have to think about it as such a large decline over a relatively short period. You talk about evolution, right?
Exactly. I mean, 50 years?
Yep. It cannot be strictly genetic.
No.
So it must be some sort of environmental component. What that is exactly, we're not necessarily certain. I study a class of endocrine disrupting compounds.
That's what we like to discuss more.
A little bit.
Yeah, exactly. Before, let's put a base, because I already see that we are going to be very interesting aspect. Yeah. So before we continue to answer some of the questions, let me ask you another basic concept. Yeah. There is always, when we talk about infertility, for centuries, we always blame women on the infertility problems. And I remember even when I was in medical school, there were a couple that would come with infertility. We will only focus on doing all kinds of exams to the woman, everything. We will never look at the male, never. What is male infertility?
Well, it's really what we focus now, which I'm hoping that my research will actually change some of this, is male infertility. We're focusing on sperm quality parameters. Okay, so this is looking at an ejaculate, and you're measuring the total sperm, you're measuring the motility of sperm, so that is how sperm moves.
Yeah.
Right, which is very important because sperm has to find its way to the oocyte, and you're also measuring morphology. So this is how sperm look. It's not uncommon to see sperm with two tails, large heads. Most sperm is
So
somewhat
in a
abnormal.
sperm, looking is important.
Yes, the looks of sperm is important,
Looks is
and most sperm don't look very good. So we'll put it that way.
Yeah.
So that's one focus on what we classically and clinically define male infertility is really looking at these World Health Organization standards or cutoffs on these three parameters: sperm concentration, sperm motility, as well as its morphology.
So if I understand correctly, what you explained to us is even though the man will be able to ejaculate and have sperms, it doesn't mean that he will be fertile.
Absolutely.
Yeah? So
absolutely,
we have to look at the quality of the sperm that a man and the number. What is the basic number that you could call it still a man fertile or not?
The concentration, so this is per milliliter of ejaculate, is 15 million sperm. So that is the cutoff, right?
So
below that, then you'd be considered infertile. Right.
See.
But one thing you always have to think about as well is semen parameters vary
I
from
heard
time
about the
to time.
semen parameters. Yeah.
Right.
Could you explain a little more? What is the meaning of looking at semen parameters? Because always what I was aware is you count the number of sperms.
Yep.
If it's too low, that's not good. If it's higher, you're okay. But semen parameters, we haven't followed much.
Yeah, semen parameters. My job as a researcher, I'm trying to move past looking at semen parameters and diagnosing male infertility by using these kind of old, archaic ways of doing this. We've been using semen parameters to diagnose male infertility for 50, 60 plus years, and things have not changed.
Mm-hmm.
However, there's been a huge, in the last 2 decades, especially in the last 10 years, there's been a huge change in the molecular toolkit that we have. And we're not harnessing any of that data to really come up with novel biomarkers or markers of male reproductive health, as well as offspring.
So let me see if I understand what you say. So we can have somebody who has the normal number of sperms and still be infertile.
That's right. That's called idiopathic infertility.
And that could be associated with the markers that you see in the semen.
That's right.
Oh, fascinating.
That's right. So idiopathic infertility is your semen parameters are normal, but you still cannot get pregnant. Okay, so idiopathic is unknown cause.
But something is wrong in the semen then.
Or the semen or the sperm that we're not capturing by these
existing
the sperm parameters.
I want to again invite everybody. If you have questions, continue putting your questions in the chat. If somebody would like also to bring a question directly, you can unmute and please join us in the conversation. Also to those who are attending directly here, because now we have things are improving, so we have also public attending our meeting. Please feel free to bring questions in the conversation. It's a pleasure to have with us tonight Dr. Rick Pilsner, an expert in male fertility and environment. And that's a subject that I think I would like to start moving forward now that we understand a lot of questions. What are the factors that will affect male fertility? And I see we have one question from the public. Can stress impact the quality of the sperm? I don't know exactly what it means, stress. Maybe the person who put the question can define more, but stress in general.
Yes, psychosocial stress in general. Certainly, there's a whole host of different environmental factors that could influence male reproductive health. And one thing I think is important to note is how we define the environment.
Yeah.
Right? So the way that I look at the environment is anything that's not genetic. So within that encapsulates, you have nutrition, right? You have chemical exposures, which is part of my research, what I focus on. But there's also societal effects as well, neighborhood effects, the access to fresh fruit and vegetables. So a lot of different things play a role, and stress is one of this component. Psychosocial stress certainly has an impact in infertility. It's been understudied in the male, but this is something that really we should be looking at.
So you were telling us is what the and still we haven't getting the conception part, but it's just in the health of the male. So what we eat, whether we smoke or don't smoke, what we drink and will have an effect on the quality of our sperms.
Yes, but the quality is one thing. If we talk about sperm quality, I would also like to bring up the idea of epigenetics.
Oh,
yes,
that's another word that we have to go to that.
Yes, indeed. And this is what my lab focuses on. We run a environmental epigenetics lab.
So
and,
yep. And so that would be more to the long-term effect in pregnancy and so on.
It could have downstream long-term effects of the health and development of offspring.
We will go to that. I still think we are in the discussion of the male and the sperms. Because I want to jump on how is the effect of the pregnancy and then the babies in the future. But I think we have another question. Please.
So, are there supplements or medications to counteract any negative environmental factors to male reproductive health?
Not that I'm truly aware of. There's been a lot of research on antioxidants. There's been a large trial in NIEHD at NIH. It's done a large trial on zinc and folic acid supplementation. From my understanding, there's been no effects on these sperm parameters.
Interesting. Yes. This is an area we know very little.
Yeah. Right. But it's not all about the sperm parameters. I want people to move away from the sperm parameters.
It's not only that. It's not so simple.
It's not as simple.
It's more complex.
Yes.
So before still we are in the sperm, does the age of the individual has an impact on the quality of the sperm?
Yes, absolutely.
Oh.
Yes. So there's a few different things that we could bring up with age. Certainly, actually, I do have a slide on aging. And it really gets back to this, what we consider the self-renewal of spermatogonia, the stem cell for sperm. It's the self-renewal that happens. And so one more, next slide, please. Right. So the spermatogonia divide every 16 days.
Yeah.
And then it's recycled. One of those two cells is recycled back.
Maintaining the reserve that we call.
Exactly. So you can imagine that if you do some calculations, that a 40-year-old man, the spermatogonia have divided about 700 times.
Oh my goodness.
And so is that a problem? Well, yes, because any kind of mutations or epigenetic errors that occur during this mitotic division of the spermatogonia can be carried forward and can accumulate. So it's been known that older men have a higher risk of fathering children with autism and schizophrenia, so more neurodevelopmental.
Okay, so that is extremely important because, again, everybody thinks that only is the oocyte of the woman. When it gets old, if she gets pregnant, then she's going to have a baby with complications.
Right.
And men, we can have babies until we are 100 years old, and the babies will be healthy, blonde, and nice looking. So you're telling me that's not true?
That is, there's a higher risk of having children with neurodevelopmental problems.
It has nothing to do with the mother.
It has nothing to do with the mother. Absolutely.
Very, very interesting. So you mentioned a lot this word, epigenetics. Could you tell us a little, what is the meaning of epigenetics? Because what we know is, we have our genes. I got the genes from my father and my mother, and all my life and the life of my children is going to determine by my genes. Just mentioning this word of epigenetics, what is the difference between the genetics and the epigenetics?
Right. So if you just take the root word epi, what does it mean? It means above. Okay, so you can think about epigenetics as above genetics. And a really easy way to explain this is every cell within our body has the same genome, right?
Yeah.
Our neurons, our liver cells, our muscle
Which
cells.
is what we got from my father and from my mother.
Right.
Yeah, that's what I mean.
Right. But our neurons behave very differently than our liver cells or our heart cells, right? But if we have the same genome,
How do we differentiate?
how do you make different cell types?
Yeah.
And so this is really what epigenetics is. There's certain tags, and I have a slide on this as well. There's certain tags that could go on to DNA. That's called DNA methylation, or the way DNA is wrapped around certain proteins called histones.
Uh-huh. So histones is what?
They're these proteins that help compact DNA.
Our DNA.
Within the nucleus.
Okay,
right.
So it's like the boxes that keep in all the genomic material.
That's right.
Okay.
That's right. So really the two major mechanisms is DNA methylation and histone modifications.
Mm-hmm.
And together they act in concert to turn genes on and off.
Uh-huh.
So that's where you get different cell types, because certain cells are expressing or have a gene on versus a gene off.
I see.
Well, the classic definition of epigenetics is changes in gene expression without changes in DNA sequence, but that's kind of a mouthful. I think the analogy of just thinking about different cell types and how they behave, your neurons behave very differently than your liver cells.
And these epigenetic modifications, what you were saying, they are affected by the environment. So now, we learned in school that we always transfer the genes to our children. What about the changes, the epigenetic changes? Do we transfer also those changes to our children?
Yep, this is where kind of the cutting edge research is going right now. We're looking at how these epigenetic marks can be inherited. And so we thought 20 years ago that it was all genetic inheritance.
Exactly.
Which it is, majority of it is. But there is a possibility to inherit different epigenetic marks that are shaped by the environment. So, an example for sperm, during spermatogenesis, which is 74 days, different environmental exposures during that time period can have long-lasting effects on the offspring through epigenetic changes.
So those adaptations that Darwin was mentioning, the size of the wings and other things, it's not only genetics, it can be also environmental that change the epigenetic modification.
Right.
But going back to the sperm, and I think this is critical. So that means that what we do in our lives, in terms good or bad, and let's go for the bad things. If I smoke, or if I live in a place where there is environmental pollution, those things may change my epigenomic. Can we call it like that?
Yes.
And then those things will lead also to affect my children.
Absolutely.
Wow.
That's what we're
So tell me a little more about your research in that area.
Yeah. So my research looks at endocrine disrupting compounds, and so the endocrine system is hormones. So there's different. It's a broad class of environmental toxicants. I study phthalates in particular. They're a class of EDCs.
I hope that the people know what is phthalates.
Phthalates.
They don't know. The public here,
I'm getting there.
Please.
I'm getting there. Please. Yes. So phthalates are a broad class of chemicals. You could find them in pretty much anywhere in your house.
Really?
There's high molecular weight phthalates, and phthalates it's a tricky word to spell. It's Phthalats. So it's a unique word. But the high molecular weight phthalates are vinyl fluorine. Any vinyl products are made almost exclusively out of
those
phthalates.
Are plastic bottles, or where do you find them?
More vinyl flooring, your shower curtain, toys, any malleable plastic can be made of phthalates as well. They've taken phthalates out of toys these days. The second class of phthalates are low molecular weight phthalates, and they act as stabilizers in perfumes and personal care products. So if you wear a lot of personal care products for men, aftershave, shaving lotion with cool breeze scent, these things, those artificial scents are stabilized by phthalates.
Oh
so
my
you're,
goodness.
you're actually putting on anti-hormones in a way,
Wow.
anti-testosterone chemicals onto your body.
And then I remember, I mean, it was very popular that you would walk in a room and every man had a different smell that it was impossible to breathe sometimes. As God, now I don't feel that is the case now. Right. So very, very, very important. We have a question here. Do we know anything about how environmental factors like air pollution or viral infection, COVID-19, patients got affect male fertility and fetal development? Because, you know, all the infections, we talk only about the mother, the mother, the mother.
Right.
What about if the man is infected, like COVID-19 or any other viral infection?
Right. Yeah, I think there's absolutely a possibility that could have long-term effects on the offspring. I do not know any studies right now looking specifically at the sperm epigenome or those contributions. I think it's still too early, right? We're still just getting out of COVID. I think that data is being collected now.
Yeah.
So it may be a few years until we get that data. Certainly, if we switch to air pollution, there's definitely a influence or increased risk if you live near closer to a highway, for an example.
Really?
To have more poor semen parameters and possibly.
Sorry to interrupt. So we see the environment will affect the fertility of the man, but it can induce epigenetic modifications that will have an impact then in the quality of the pregnancy, for example, or in the health of the baby.
Right. There's two aspects of that. Yes.
Yeah.
Does it influence reproductive success? Is someone having a live birth? Yes or no? That's absolutely critical. But then the future, the trajectory could really offset the trajectory of normal health and development of the offspring. So I think this is kind of an important aspect of it's not about just the mother. It's about the father and how to get a healthy sperm.
So, you know, again, my training as OB, we always thought that all the pregnancy complications is because either the mother was doing something wrong. Yeah. But what you're telling me about, well, at least extracting from the information that you're giving us, is that maybe some pregnancy complications are not only due to what the mother was doing, maybe also what the father contributes.
Oh, absolutely. And again, when I started this talk out, it takes two to tango.
It takes two to tango.
And if you think about the process of spermatogenesis, that it takes 2.5 months.
Yeah.
Men have an environmental responsibility too, prior to conception, and that's where I think we think about the health of the baby only after conception, and we now know that the preconception period is the earliest important
and it
period.
can be as critical for the success of the pregnancy.
Absolutely. So men need to step up. They need to have an environmental responsibility, and it starts at least 2.5 months prior to conception. At least, but if not even more. So
because I have seen about friends, I can smoke, I can drink, I can do whatever I want, reproduce, and that's my problem is my responsibility is gone.
Right.
We have been doing a major damage.
Possibly, absolutely. It's certainly the cards are there. For sure.
So we have a question here. Let's see if we can go. There's a long question. Hormonal stress, cortisol, adrenaline, slows the growth of embryos and the stem cells in the lab, in the petri dish. Environmental biochemical stressors also slow growth. Together, the two types of stress synergistically slow growth. Are there similar synergistic effects at the level of the spermatogonia? You will have to explain a little what is synergistics, and of course in the spermatogonia.
Right. Well, synergistics is the combined effect that not additive, just not 2 + 2 = 4, but maybe 2 + 2 or 2 × 2 is 8. Something like that, right? So there's different hormonal stress, right? The question is getting back to slowing the growth of the embryo. One really key finding from my lab is going back to the phthalates. We've showed that preconception phthalate exposure in males, irrespective of the female exposures, influences embryo quality.
Wow.
At day 5. So the blastocyst quality, which is then, my study was through an IVF clinic. So we have these very detailed morphological characteristics of the embryo, but we show that male exposures to these phthalates can influence the quality of the embryo.
Even if the mother is healthy, is
right.
isolated from the exposure of that, if the father was exposed, it will impact the baby.
Right. Well, we controlled for the levels. Right. We controlled for the levels of the female phthalates as well. So that's a very important aspect of this. And within that same study, we showed that the same phthalate metabolites. These are anti-androgenic phthalates.
Anti-androgenic. Okay.
Yes.
Explain a little bit meaning of the anti-androgenic.
What are androgens, right? Androgens are, you think about testosterone, right?
Are male hormones.
Right, which are very important for spermatogenesis. Yeah. But certain phthalates are considered anti-androgenic.
So will inhibit
they reduce testosterone levels. So these phthalates exposures that we had in males influence sperm epigenetics, and then also downstream influenced embryo development. And when we looked at which genes that the epigenome was changed based on these phthalate exposures, we found that most of the genes were in pathways for early life development. So it made a lot of sense.
So the early stages of the development of the embryo, the formation of all the organs of the future baby, are determined for the health of the male. That's what you're telling us?
They're contributing. The male is contributing
to it has an important role.
Absolutely.
So whatever we did before the conception will impact how our baby is going to develop.
That's right. That's right.
Fascinating. So that is when exactly, I think you covered one of the questions that I have here, how the healthy sperm contribute to a healthy baby. I don't know if you want to add something else in this, or you think we have covered this aspect.
Yeah, I think we've covered it pretty well. Men have an environmental responsibility. You need to get your sperm into shape before you start conceiving, right? So planning a pregnancy, I think, is very important as well.
Fantastic. So we age. We talk about aging and so on. Is the same thing our age, our chronological age, or the sperm age? What is a sperm age?
That's a great question, and there's been a movement in the last 5-10 years of not thinking about age as chronological age, right? We think that 1 year is one orbit around the sun.
Yeah.
Does that really predict disease, right? Because you can imagine all your friends that you went to high school with, they're the same age.
Yeah.
But people get different diseases at different times.
It's true.
So is it just chronological age, or is there a better way to think about age? And what I think about age is more biological age. So not chronological age in a way, but how old your cells are. And I think that's a very important concept, is we emphasize on chronological age, but what is captured in biological age that's not captured in chronological age is your lifestyle and some genetics as well. So if you are
epigenetics,
well, that's how we measure it. We'll get to that in a bit. Okay. But you think about someone who smokes, who drinks often, that has a very poor diet. Their biological age is likely mismatched with their chronological age. Meaning, if they are 35 years old, their biological age maybe is 40.
Oh,
and so we've actually research that we're ready to publish and put in a provisional patent for, we've looked at sperm aging, and we've come up with a algorithm that can calculate your sperm age.
Wow. So in other words, now I was generalizing that you have a 60 or 70 years old man, and I thought, okay, those sperms that are old, that is not good. But if you're telling me you can have a 70 years old or 60 years old man, and maybe also have a sperm age that is young or healthy based on what he does.
Right.
Or the opposite.
Absolutely.
A 60-year-old may have an 80 years old sperm age.
Right. Right.
With the consequences of both.
Right. Or more importantly, those folks that are in reproductive years that are trying to have families, those in their 30s, say, for example, their sperm may be 40 years old, maybe a decade,
or 50 years old,
or 50 years old,
which is not good.
And what we found in our study is that those men that have an older sperm age, that means older than their chronological age by 1 year or more, have almost a 40% reduction in reproductive success, in getting pregnant.
Wow.
So this is not accounting for anything measured in the female besides female age and BMI. We're not looking at the reproductive
BMI,
history.
you mean the weight of
right, the weight, yes. Yeah. So we're not looking at any of the female reproductive history of the women, but we have this marker, this sperm age, that could predict reproductive success very strongly.
Impressive.
Right.
So the age can. So how do we keep a young sperm?
Exercise, nutrition.
Don't tell me diet, because my wife now is going to even stress that she have to keep a diet when I was younger.
Right. it always reminds me of one of Michael Pollan's books. That's, when we go to the grocery store, we want to stay always on the outside of the grocery store. This is where the fruit is, the vegetables, the eggs, the milk. All those aisles in the middle are processed food and canned food and things. So if we have a healthier lifestyle, that likely is
avoid the processed food.
Right.
But the meat is okay.
Wow.
Don't answer that question.
Like everything else.
Excellent. How are we doing with time? I think we are fine. We have still a few questions coming there. Are environmental factors affecting genetic or epigenetic changes? I think we mentioned a little, but it would be great if you can define and differentiate what are the effects in terms of the genetics and what are the effects in the epigenetic. How do we differentiate that?
Right, and it goes back to the self-renewal of spermatogonia. If there's these epigenetic, mutations that occur, they will accumulate over time. And then that means that the next generation of sperm that's being produced 16 days later, they are all harboring that genetic mutation. So these mutations will accumulate over time. That's why older fathers are at higher risk for having offspring with different complications. Exactly, and the same is with the epigenome as well. And this idea of this sperm age, I think, could be a novel way to look at reproductive success.
Let me go back to the sperm age, and sorry, I jumped to the factors. How do you determine the age?
Right. So
there is a trick in that, or
yes, it's all magic. It's all magic. Yes. So there are certain regions that we know that change the epigenome with age. Okay, and this is sperm specific. So it's not for all the other cells in your body, but just for sperm. So we take those areas or those markers, these epigenetic changes, and we throw them into a computer algorithm system. I have a collaborator, Laura Balzer. She's a biostatistician at University of Massachusetts in Amherst that has really driven that aspect, the computational side. And what it does is, out of our 10,000 potential markers, it picks 140 or so that best predict chronological age and these environmental, reproductive outcomes.
So if I understand what you're telling us is that you can take the made up of the specific sperm, and then based on what is expressing, what is the content, you can determine if it's a young sperm, a mid-term sperm, or an old sperm.
Right.
Yeah.
Exactly. And this, I think, is really interesting because this could offer couples an easy way to inform their course of treatment if necessary.
Yeah.
If couples are wanting to get pregnant immediately, this could be an opportunity for them to be informed on, well, maybe we should get reproductive assistant technologies and go through IVF instead of trying to reproduce naturally.
Yeah. So the other aspect that we have of us is those environmental factors. Well, we covered the capacity of the reproduction, but you mentioned. Let's go now. Let's move to the part of when Mars meets with Venus. Yeah. When the male meets the female. What is the role of the female reproductive organ in terms of the activity of the sperm?
Okay. So there's certain hormones that are within the female reproductive tract that actually stimulate what we call hyperactivation and capacitation of sperm.
Oh.
So they make the sperm tail hypermodal.
So to start swimming
faster and faster and faster.
What's the competition? The race starts.
Right. That's it. So
and that race is activated or initiated, you say, by factors produced by the female.
Female, absolutely. So that's where that's critical component of natural conception is the female interaction with the sperm as well. And we talked about seminal plasma. Another role of seminal plasma is inflammatory markers that kind of give a signal to the female reproductive tract of, well, this is a foreign cell, but let's not attack it and kill it. It's maybe here for a beneficial reason, and we should welcome the sperm in versus having more of an immune response and thinking it's a foreign entity.
And that's what we discussed, I think, in one of our meetings, that the baby is a foreign body, but really the whole recognition starts when the sperm comes into the female reproductive tract. But also, the uterus needs to be wake up or activated to have the baby.
That's right. Well, you call that kind of priming in a way.
Priming of the uterus. And that you say
plasma
will be the semen.
Yes.
And the signals coming.
Absolutely.
This what you call this extracellular vesicles. Yeah.
Yes.
Do you think they have a role in this priming of the?
I think so. The research at this component, at this stage, again, this research is in its infancy. That people are just looking at how it affects sperm and embryo development within that small context, but no one has really looked at extracellular vesicles in seminal fluid and how it contributes to, say, an immune response.
We should look at that.
Hey, you're the
exactly.
expert.
We're doing the immunology. That's what we do for a living. We have to talk about this after this meeting. Okay. 30 million sperms, 15 million sperms have been delivered in the vagina of the female reproductive tract. It starts going up. One is the only one.
Winner takes it all.
What it makes that? How do you control that from 30 million? It's only one that will do the job.
That's how nature rolls. It's a hard drive getting all the way up there. And
so there is a selection process. Yes, evolutionary, you're selecting for the best one?
The one that has, yes, the sperm that is in the best shape.
Best shape.
We'll put it that way. But not just morphology or morphologically, but also you have to think about the signals within that epigenome. That's, I think, contributing to this as well.
So that brings me to now a more I don't know how to say, ethical question and so on. When we deal with infertility, and when we have a patient, a male patient who's infertile, still is producing sperms, and you're correct, a low number. And we take those sperms and we do something that's called ICSI. Mm-hmm. So So we just pick one sperm and we introduce in the oocyte. What are we doing?
That is getting away from the sperm race, right?
Exactly.
Yeah. So,
I mean, we are not selecting. I mean, there is methods to select the best sperm.
they certainly isolate sperm with the best motility and try to.
But you tell me that doesn't mean it's the best sperm.
That's
so
right.
We're guessing in a way.
That's right. So that's where we need to look at our molecular toolkit and see if
that's when your aging will be very important.
Right. So I think there's needs to be an advance in how we view healthy sperm, and it's just not how it looks. It's not how it moves or how many we have.
Yeah. So we have a long way in order to help the male fertility. So ICSI really is not. I'm going to get in trouble with what I'm going to say. You correct me. So it is a fantastic technology that you can pick one sperm and put it inside of the oocyte and then to do the IVF transfer. So we are breaking the rules of nature, that is the natural selection.
We've been doing that for hundreds of years with farming and picking that one plant that is producing the biggest, best tomatoes and selecting. So, but in that,
so there is another steps that will do the selection if we did something wrong, pick it, or if nature by accident, the winner was a bad one.
Right. That could be problematic.
That could be it. But there is potentially in the different stages of the embryogenesis or the development of the embryo, when nature will determine whether things are going well or not.
Right. And I think that we've gained a lot of insight with IVF technologies and using ICSI, that we could kind of monitor how embryo development, that process is going, and hopefully that translates into a healthy baby.
Fascinating. I think we learned a lot tonight. At least I learned a lot, especially this aspect of the aging, of the sperm aging is fascinating. Congratulations. So we had an incredible evening learning about the role of the man in the preconception and its impact on the health of the pregnancy, first of all, but also in the future health of the individual. It's not only the genes that we are passing, the genes that came from our fathers and from generations, is also how our behavior before preconception is going to affect the quality of the genes that we are going to transfer, those epigenetic modifications. So if I can summarize all what you taught us tonight is we have as males a responsibility for generations to come, for the good and for the bad. So how we prepare ourselves in terms of maintaining the male health before the preconception and the preconception life will have an impact not only in our children, but in the children of our children, generations down the road.
Absolutely.
I want to thank you again for joining us tonight. It was a fascinating evening. And I hope also everybody who assisted enjoyed and learned the subject. To be continuous, there will be new things that we would like to expand. But I think for tonight, we want to just introduce the general concept.
Great. Thank you for having me.
Thank you. And thank you, everybody, for joining us. Again, let me remind you that this session has been recorded and it will be presented in a podcast, as well as it will be available in media, Facebook, and Lisa, what other? And YouTube. So if you miss or any of your friends would like to follow this presentation, they can go to the media and look at it. Thank you very much. And let me remind you that we will not have the activities or the Meet the Expert during the summer. We want to wish you an enjoyable summer. I think after the pandemic, this is the summer that everybody wants to go out to the beach. So I recommend you and be healthy, exercise. We will coming back in the month of September, that is the month of ovarian cancer. And we will discuss about ovarian cancer and meet the expert in the month of September. Thank you very much again.
Thank you, Dr. Moore.
We'll see you later.
All right.